Yeah, so a lot of the rationale comes from observations that we made that tumors that have a robust immune infiltrate in them prior to the initiation of preoperative or neoadjuvant chemotherapy actually do better and respond better to treatment and have better outcomes. And so what we were looking for were what types of drugs or treatments could we use in the triple negative early non-metastatic setting to convert more of the tumors that were relatively immune cold or did not have a robust infiltration into those that did have a more robust, inflammatory signature. And oncolytic viruses are ideal drugs to be able to kind of provoke that type of reaction in the primary tumor. And that's where the idea was born about trying to combine oncolytic virus with preoperative chemotherapy. And this particular trial that we're presenting was initiated prior to the advent of KEYNOTE-522 as standard treatment. So we actually just did oncolytic virus with standard anthracycline taxane chemotherapy at the time, which was standard for triple negative breast cancer. And we found that the outcomes that we're presenting at this year's ASCO meeting are actually quite comparable to KEYNOTE-522. Small trial, however, the results were quite encouraging to suggest that the oncolytic virus could boost outcomes beyond what we would have expected with traditional chemotherapy alone. There's been a lot of debate in the community specifically about the linkage between pathologic complete response rate as an early surrogate endpoint and long-term outcomes for women with breast cancer. And it hasn't been uniform across the board. So for example, hormone receptor positive breast cancer, the linkage between PCR status and long-term outcomes is a little bit less well defined. Whereas for triple negative breast cancer in particular, it does appear that there is quite a strong correlation between hitting that PCR mark, for example, or not, and how well the patient's likely to do in the future. And so it gives us an ideal window, if you will, to use PCR in the triple negative setting to vet new agents and treatments more quickly than just waiting for the long-term outcomes. And so I think that's why triple negative is an ideal situation to be able to test the PCR endpoint for long-term outcomes. Well, the data was quite interesting, though. We found that, in essence, it reinforced and supported our underlying hypothesis that if you're able to get a more robust immune expansion or infiltration over time when you're doing a therapy, that you are likely sensitizing the tumor to the preoperative therapy and getting better response rates. And that's indeed what we saw on the TVEC trial, was that those patients that went on to ultimately get a PCR appeared to have larger expansions of certain T-cell subtypes and also, interestingly, B-cell clones as well. And so there may be a role whereby using an agent like an oncolytic virus can actually instigate a cooperative cycle, if you will, between B cells, which can actually present antigens similar to other immune cells, and the surrounding T cells, and they can form these ectopic lymph node structures, we call it, that is almost like a forward operating base for your immune cells right at the verge of the tumor. And so that may provoke a more robust anti-tumor immune response in those settings. So I think, again, it supported our hypothesis that this is a favorable thing to do and that it's likely associated with a better response to preoperative therapy. The safety profile for the incorporation of TVEC with standard chemotherapy was excellent. That was one of the things, though, that we did have to address with the FDA when we opened the trial, was that we had to monitor patients for potential reactivation events because some of these patients may be somewhat immune compromised during the period where they're getting chemotherapy. But that fortunately was not seen. We did not see any kind of major reactivation issues in the long-term surveillance following the chemotherapy. And the toxicities that we observed in combining it with chemo did not show any new safety signals or unexpected events. So it's a highly feasible, manageable approach to be able to boost the response to therapy. And also, it didn't necessarily increase some of the autoimmune toxicities that we have some concerns about with other strategies that women now may have to deal with, in the aftermath of treatments down the road with their adrenal glands or their thyroid. So again, I think it was a very favorable toxicity profile and bodes well for us to continue to explore it in the future. Yeah, we're actively looking and exploring with different partners that are developing various oncolytic agents that are looking at the next generation of viruses that are armed with payloads and other immune stimulating constructs to be able to get even a more robust reaction at the tumor site where you're injecting. And so we feel that the future is going to be an intratumoral response and treatment paradigm, if you will, that will allow us to incorporate these agents on top of, say, the modern Keynote 522 backbone, whereby we can further boost the complete elimination rate of these tumors and cure rates without increasing the systemic toxicities significantly for these patients. And that's really, I think, where we want to go with this approach. And hopefully we'll be able to recruit some of these stakeholders that have novel oncolytic viruses into doing some of these next generation trials.